Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome to the deep dive. Our mission is, well, pretty simple, giving you the fastest possible route to mastering complex concepts.
And today we are really getting into it.
We're diving deep into what is, I think, the single most crucial biological equation on Earth, photosynthesis in higher plants.
Absolutely.
Now, this is foundational science, and we're pulling it from key sources, but we're framing it specifically for those of you preparing for those really knowledge-intensive exams. We're going to be linking the core biology to the general knowledge facts you just have to know. And before we unlock the engine of life, I just want to take a moment to acknowledge the foundation of this work. A huge, massive thank you to all of our existing members.
We really appreciate it.
Your support is what makes creating this super useful content possible. If you find this knowledge helpful and, if you're capable, please consider subscribing and joining the paid membership on YouTube.
It really does help.
It ensures this content keeps flowing to you. And also, just take a quick second to this deep dive and share your valuable comments. That engagement, it really fuels our research.
It does. Okay, so we are examining the fundamental process of carbon assimilation.
Right.
Think of this as tracing the path from raw external energy sunlight all the way to synthesized sugar. I mean, the organic matter that supports pretty much every food web on the planet.
And we'll hit the historical discoveries, the specific chemistry, and some of those evolutionary adaptations that make it so much more efficient.
Exactly.
Okay, let's unpack this then. When we say photosynthesis is a physicochemical process, what do we really mean?
Well, it's acknowledging that it's not just a plant sort of passively soaking up rays.
It's an active factor.
It's an active factory. It's part physics capturing light and part chemistry building molecules. It's the primary source of all food, yes, but crucially, it's responsible for the ongoing release of the oxygen, the O2, that shapes our entire atmosphere.
The scale is it's stunning. But the understanding of it, that started really small, right? With observation.
Oh, yeah. We have to go back to Joseph Priestley in 1774. He's the same scientist who discovered oxygen. He set up this famous experiment with a bell jar.
Ah, the classic setup.
Yes.
A burning candle or a small mouse sealed under the jar.
And they both use up something vital in the air, and eventually the candle goes out, and the mouse, well, it suffocates.
They were, in his words, damaging the air.
Exactly. But here's the breakthrough. When Priestley put a little mint plant inside that same bell jar, he found that after a while, the candle could burn again.
Yeah, the mouse could survive.
And the mouse could survive. He proved that plants restore whatever it is that breathing animals and burning candles remove from the air.
So he basically discovered the recycling loop of life.
In a way, yes.
Yeah.
But he didn't know how the plant did it. He just knew it happened. That's where Jan Ingenhousz comes in. Ingenhousz built on Priestley's work, and he showed that this whole process, this purification, is totally dependent on sunlight.
So in the dark, nothing happens.
Nothing. And he also, very crucially, noticed that it was only the green parts of the plant that did this. When he put them in water in bright light, he saw these tiny little bubbles coming off them.
Oxygen.
Oxygen. So that narrowed the focus immensely.
So the site of action is key. And that site is the chloroplast.
Absolutely. The chloroplast is the engine room. It's all about its internal compartments. You have the grana, which are these stacks of thylakoids.
Always picture them as stacks of coins.
That's a perfect analogy. And that's the stage for the light reactions, where the light energy is actually captured. Then you have the stroma.
The fluid matrix?
The fluid all around the stacks. And that fluid is the site of phase 2, the dark reactions, or what we now call the carbon reactions. The physical separation is just key to efficiency.
Okay, that separation leads us perfectly into the next bit, the energy conversion phase. The light reaction. What's the big picture job here?
Its core mission is to convert that fleeting light energy, the photons, into stable, usable chemical energy.
So we're talking about molecules.
We're talking about molecules, specifically ATP and NADPH. Think of them as the high-energy currency. You bank the energy from the light phase so you can pay for the food-building phase later.
Right. And to catch that light, the plant needs very specific tools. I mean, we see plants as green because they're rejecting green light, but they're absorbing like crazy at the two ends of the spectrum.
Yes. The absorption is most effective in the blue and the red regions of light. Chlorophyll is the indispensable primary pigment. It does all the heavy lifting.
But it's not alone, right? There are others.
Not at all. We shouldn't forget the accessory pigments like chlorophyll B and the carotenoids. They act like antennae, grabbing energy from other wavelengths and just funneling it all back to chlorophyll A.
Okay, but hang on. If they have all these accessory pigments, why even bother with chlorophyll as the main hub? Is it just, I don't know, an evolutionary thing?
That's an excellent question. Chlorophyll sits at what we call the reaction center because it's the only molecule that can actually perform the charge separation.
What does it mean?
It can take that energy and literally launch an electron into the next phase of the process. The accessory pigments just gather the energy. Chlorophyll is the one that converts it into chemical action.
And that action is visualized by the Z-scheme. It sounds complicated.
It does, but it's really just a roadmap. It's tracking the journey of an electron.
Okay.
It's the roadmap for what we call non-cyclic photophosphorylation. It shows the electron flowing through photosystem 2, or PS2, and then over to photosystem I, PSI.
And the start of that journey is the splitting of water, right? Photolysis.
And that is fundamentally critical for all life on Earth. Water splitting, H0 is split, providing 2 electrons to refill the gap left in PS2. And as a world-changing byproduct, it releases oxygen.
That's it. That's the source of all atmospheric oxygen.
A single enzyme-driven reaction. The electrons then move through a transport chain, losing a bit of energy, which is used to make ATP.
Through a process called photophosphorylation?
Which is just a fancy way of saying making ATP from ADP using light.
Got it. And the electrons end up at photosystem I, where they're used to make NADPH.
Exactly. So we have our products, we have ATP, and we have NADPH, our high-energy currency.
So now we take that currency into the stroma for the dark reaction.
We shift into the biosynthetic phase. Now, we call it the dark reaction, but it's important to remember it is completely 100% dependent on the ATP and NADPH we just made in the light.
Of course. No light, no currency, no building.
Exactly. This is where the K-euro from the atmosphere is finally fixed into stable sugars.
And the map for this whole process came from Melvin Kelvin.
He used radioactive carbon-14 in algae, which was brand new technology at the time. It won him the Nobel Prize in 1961.
I can't imagine how difficult that work must have been, tracing a single carbon atom through a whole cycle.
It was absolutely pioneering. He mapped what we call the C3 pathway, or the Calvin cycle.
Right.
In this cycle, the Acor molecule gets attached or fixed onto a 5-carbon compound called RuBP, and the very first stable thing it makes is a 3-carbon acid.
PGA phosphoglyceric acid.
That's the one, and hence the name C3 pathway.
And the enzyme that does this, the one doing the fixing, it's a pretty big deal in the biological world.
It is the single most abundant protein in the entire biosphere.
Wow.
It's called Rubisco, which stands for ribulose bisphosphate carboxylase oxygenase. And that name, the name itself, hints at a huge inherent problem.
Okay, let's break it down. Carboxylase. Good. That means it fixes carbon dioxide.
Correct.
But oxygenase, that sounds like it can also bind to oxygen, which seems bad for a process that's supposed to be about carbon.
That is the evolutionary accident we're dealing with. In C3 plants, which do this whole cycle in their mesophyll cells, when CO levels drop and oxygen levels rise...
Like on a hot, dry day when the plant closes its pores.
Exactly. On those days, Rubisco starts acting as an oxygenase. Instead of fixing carbon, it binds to oxygen, and that kicks off a really wasteful process called photorespiration.
And wasteful is bad because it uses up the very ATP and NADPH we just worked so hard to make, but it doesn't produce any sugar. It's a massive hit to efficiency.
A huge hit. And this brings us to this fascinating evolutionary leap we see in some tropical plants. The C4 pathway.
The Hatch and Slack pathway.
That's it. And this adaptation is designed specifically to suppress that wasteful photorespiration.
So what's their trick? It's about separating things in space, right?
Yeah.
Using 2 different kinds of cells.
Exactly. C4 plants, like maize or sugarcane, they start by fixing co-euros in their mesophyll cells, just like C3 plants. But, and this is the key, they don't use Rubisco there.
Okay, so what do they use?
They use a different enzyme. The primary co-euro acceptor is a 3-carbon molecule called PEP, phosphonol pyruvate. This fourth step creates a 4-carbon acid.
OAA, oxaloacetic acid, hence C4 pathway.
You got it. And this first enzyme, PEP carboxylase, has a much, much higher affinity for co-curos than Rubisco does. It's a co-euro magnet.
So they grab the curo outside the main factory and then they shuttle it inward.
That's the brilliant part. The OAA is transported into these specialized cells that surround the veins, the vascular bundles. We call them the bundle sheath cells.
And this is where Rubisco is waiting.
This is where Rubisco is waiting. The mesophyll cells have no Rubisco. The bundle sheath cells are where the real Calvin cycle, the C3 pathway, actually happens.
So it's like a chemical pump. The OAA gets in there, drops off the Chiaro, and creates this super concentrated Chiaro environment right around Rubisco.
Which means that inside that special cell, the concentration of Chiaro is so high that Rubisco is basically forced to act only as a carboxylase. It ignores the oxygen. Photorespiration is virtually eliminated.
Which is why C4 plants do so well in hot, dry, sunny conditions. They've engineered a way around Rubisco's big flaw.
They've engineered a workaround for its Achilles heel.
That dual cell mechanism is definitely a high-yield concept.
Mm.
Okay, let's connect this foundational science to some broader knowledge points, drawing from the sources.
Let's do it. Let's tie this back to minerals. We talked about water splitting and oxygen evolution in the light reaction. Well, that incredibly important step is catalyzed by a metal ion.
Manganese.
Manganese. It is absolutely essential for that water splitting complex. It's a direct link between mineral nutrition and, well, the air we breathe.
And on the topic of critical elements, there's always nitrogen.
Always. It's the most prevalent element in living things. Plants need it to build amino acids, proteins, like Rubisco itself.
And how does it get around the plant?
Well, some nitrogen travels as inorganic ions in the xylem, but the majority of it, the really critical building blocks, are transported in an organic form as amino acids and related compounds.
So the plant moves finished parts around, not just raw materials.
Much more efficient.
And getting that nitrogen in the first place relies on some very specialized life forms. The whole process of nitrogen reduction, taking N from the air and turning it into usable ammonia, NHAs.
That's done by the enzyme nitrogenase, and it's only found in prokaryotes.
Bacteria and their relatives.
Exactly. This is a huge ecological concept. You have free-living nitrogen fixers like cyanobacteria, things like nostoc and anabina. They're doing this vital service all over the world.
And our source material confirms the practical application here. Nostoc is actively used as a biofertilizer that connects microbial ecology directly to sustainable agriculture.
And that is definitely tested knowledge.
Okay, finally, let's just briefly touch on how the environment shapes the plant itself. In really dense places like equatorial forests, the whole architecture is driven by the race for light.
Absolutely. The presence of these tall, closely set trees forces a unique growth pattern. Their crowns, their tops, form a continuous, tight canopy.
To maximize light capture?
It's a physical adaptation to maximize that very first step of photosynthesis in a super competitive environment.
So we've traced the entire path of carbon assimilation, from those first historical experiments that figured out air in plants, through the complex Z scheme, all the way to sugar synthesis in C3 plants, or with that clever C4 workaround.
A complete journey.
So what does this all mean? The primary job of photosynthesis is to trap light energy and store it in chemical bonds. It's the planet's solar battery. But if the plant has made all this food, why does it also need respiration? Why can't it just, coast on that?
Because energy stored is not energy used. Life's ability to actually use that stored energy depends completely on respiration.
The process that breaks the bonds.
The oxidation process that breaks those carbon bonds, that energy has to be released in a controlled way as ATP to fuel growth, maintenance, water uptake, every single thing the cell does.
And here is the provocative thought for you to carry forward from this. Think about the greenest, most vibrant leaf you can imagine. Even in that leaf, not every cell photosynthesizes.
Right, only the cells with chloroplasts do.
Which means that every single plant, even the primary producers, is constantly in a complex negotiation. It's synthesizing food in one area, the chloroplasts, and consuming it somewhere else via cellular respiration just to stay alive and grow.
It highlights the absolute ubiquitous necessity of cellular respiration, running silently, constantly, right alongside the world-powering engine of photosynthesis.
Thank you so much for joining this deep dive into the fundamental physics and chemistry of life. If this content helped you connect some of those dots or clarify those high-yield concepts, please show your support.
Please do.
Like this deep dive, share your valuable comment, and consider a subscription or a paid membership on YouTube to help the team keep creating these focused resources. We truly appreciate and thank all our members. Until next time.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.